Good afternoon, everyone. Welcome to the very last in our October and Beyond session this week with our continued collaboration theme. We've been so glad to welcome you to Framing the Future in our first week, examining change, research and data, workplace tools and knowledge mobilization, and now today with continued collaboration. And we would invite you to go back if you missed any of our sessions and view the videos, and the slides will be there for your usage with your joint health and safety committees, and really just thanking you for joining the Occupational Disease Action Plan and being fully part of prevention of OC disease. So with that, I would like to introduce our first speaker. Paul White obtained his BSc in microbiology and immunology and a PhD in environmental toxicology from McGill University, my alma mater in Montreal. His PhD research investigated the sources and fate of mutagens and carcinogens in the St. Lawrence River system. He conducted postdoctoral research at US EPA facilities in Rhode Island and North Carolina, investigating the mutagenic hazards of municipal wastewaters. He joined Health Canada in 1999 and is currently a senior research scientist and leader in the Genetic Toxicology Lab group in the Environmental Health Science and Research Bureau. In 2002, he was appointed professor of chemical and environmental toxicology in the Department of Biology at the University of Ottawa. His current work investigates the mutagenic and carcinogenic hazards of complex mixtures. We know we've been talking much about this over Octobers of past and supporting Paul Demers in his recommendations around this. So the design and validation of in vitro and in vivo tools for genetic toxicity assessment, the use of molecular biomarkers for mutagen exposure assessment, and in the development of qualitative methods for interpretation of genetic toxicity dose response data. He has published over 100 peer-reviewed scientific papers, and his work's been cited over 5,000 times. From 2007 to 11, he served as editor-in-chief of Environmental and Molecular Mutagenesis, Mutation Research, and Food and Chemical Toxicology. His work has been recognized by Scientific Leadership and Research Excellence Awards from the Environmental Mutagenesis and Genomics Society, to the Deputy Minister of Health of the Government of Canada, and the Governor General of Canada. He is the current president of the International Association of Environmental Mutagenesis and Genomics Societies, and program co-chair for the 13th International Conference on Environmental Mutagens, which will be held in Ottawa in September of 2022. So please welcome Dr. Paul White.
Thank you so much, and thanks so much for the invitation to present here this afternoon. It's a bit embarrassing that you read all that. I would have provided a lot less if I knew you were going to read it all. Yeah, it's fine. So I changed my title a tiny bit, and I took Ottawa out because the work, well, what I'm going to talk about pertains to firefighters really in a variety of places. I work in Ottawa. Caraton, who I'll show you a slide about some of his work that's going on. He works with Montreal Firefighter, firefighting organization, and we have some literature review results that I'll present very briefly. So all that to say, the title of my talk is Occupational Exposures of Firefighters to Toxicants of Concern. And I'm going to be talking about collaborative work involving University of Ottawa, Ottawa Fire Services, Health Canada, University of Toronto, Université Laval, the INSPQ, University of Copenhagen, and McGill University. And I won't read all the names here of the people who are involved, except to say that I'm involved from Health Canada, along with Rocio Aranda Rodriguez and Caraton Kubwabo, who are chemists at Environment Canada, at Health Canada, and they're on the call here today. So just by way of introduction, I think this group is well aware of the fact that firefighters' risk of chronic diseases is elevated. So for example, cancer, that's my primary interest, is genetic effects and carcinogenesis. Many studies have shown that there's significant increases in cancer incidence, up to 6-fold, and mortality. And just very briefly, I have some stats here from 2 papers I list down at the bottom, Ma et al. And Daniels et al., with stats from Florida and stats from San Francisco, Chicago, and Philadelphia. And I won't read all the data here, but it shows you that the standardized incidence ratio and standardized mortality ratio is elevated. And as I mentioned up here, it goes up to about 6-fold for incidence and 2-fold for mortality. And this is obviously controversial. And the International Agency for Research on Cancer classified occupational exposure as a firefighter as only possibly carcinogenic. And usually I've sat on a lot of IARC monograph working groups, and usually when it's possibly or potentially carcinogenic, that sort of ruling, it's generally because the human epidemiology data are not very good, especially exposure assessment. So IARC has prioritized reassessment, and there will be a reassessment monograph meeting in June 7th to 14th of the next year, 2022, and it will be IARC monograph 132. So we'll see if the data, the human epi data have improved. It could go up to group 1, which would be known human carcinogen. So we started working on this in about 2015, and this is one of the papers that we published here in Environmental Science and Technology. And we worked on exposures to PAHs, metal, and organic mutagens during emergency fire suppression. And this is significant because there's a lot of literature on firefighter exposures to a variety of toxicants, but very little on shift emergency fire suppression of municipal structural fires. And the main reason is because it's not easy to do. This first study that we did, that was a McGill, Université Laval, Ottawa Fire Service, Health Canada study, was funded by the Ontario Ministry of Labour. And these are the partners who were involved. Oh, I forgot to mention University of Toronto as well. I'm just going to get rid of this here. Oh, somehow, oh, there we go. Oops, now I'm out of presentation mode. Sorry about that. I'm just having a hard time getting rid of the little thingy at the top here. There we go. So with respect to that study, we worked on, I'll just give you a rundown on the study samples that we worked on. So one thing I'll mention is all of these photos on all of my slides, they're not stock photos that come from somewhere online. These are all photos of fires where we collected samples. So fires that were in Ottawa and where we were able to collect samples from the firefighters during fire suppression. So we worked on 29 paired pre and post fire dermal wipe and urine samples collected at 19 fires with matching what we call active air samples. So these are air samples collected with a pump on each person. So we call them personal active air samples. And we also, for point of comparison, we also collected samples from Ottawa Fire Service office workers. And then moreover, we use CHMS, Canadian Health Measures Survey data, as additional controls. So all the firefighter samples were compared to these controls. And you might notice that it's a relatively small number of paired samples, but this is not easy to do. And we're at the mercy of fire incidents. So we can only collect samples when the firefighters are called to go to an emergency structural municipal fire. So with respect to what samples we collected, at the beginning of the shift, we collected skin wipes from forehead, neck, and wrist. We collected undergear wipes, so from the undershirt and underpants, and we collected personal protective equipment wipes from the jacket, belt, and pants. And then in addition, you can see up here, we collected urine samples at the start of the shift. We also, as I mentioned, collected personal air samples. Here you can see a tube that goes down the inside of the bunker gear jacket, going to the pocket, and there's a pump in the pocket, and on the back of the firefighter's neck is a sampling tube attached to the bunker gear with Velcro. And what they had to do was remove this cap and start the pump when they arrived at the fire. Sometimes they forgot to remove the cap and the pump failed, but that's another story. So here's the sample collection tube at the breathing zone of every firefighter. And after the fire, we collected an 18-hour integrated urine sample, skin wipes, PPE wipes again, undergear wipes. So basically the same here that I show on the left. And they all had to fill out a post-fire event questionnaire. So this is basically the same as what's shown here on the left. So skipping right ahead, quickly ahead to the results of this first study, we found significant increases in post-suppression surface contamination. So you can see here in the skin, this is PAH concentration in nanograms per square centimeter, pre-fire and post-fire. We found a significant, on average, 3.3-fold increase in contamination on the skin, pre versus post. On the clothing, the undergear clothing, we did not find any significant change. So probably indicative of the fact that the bunker gear, the turnout gear, is protective. And then in addition, not surprisingly, we found an average of 5.2-fold change in contamination of the PPE. So the bunker gear jacket, and this is a composite sample from the jacket, belt, and pants. So we, as I mentioned, we took urine samples. So we monitored polycyclic aromatic hydrocarbon exposure, so known, some of them are known human carcinogens, by looking at the urinary concentration of pH metabolites. This is showing only for 1-hydroxypyrene. So you can see here the level of 1-hydroxypyrene in Canadian non-smoking males from the Canadian Health Measures Survey, Ottawa Fire Service office workers, and then firefighters before the fire and firefighter after firefighting. And you can see an average of 3.7-fold increase, a significant increase. And we found up to almost 40-fold difference between pre-fire and post-fire. And I didn't show here, but we found similar results. I didn't show a plot, but for hydroxyphenanthrenes, 5.3 up to 63-fold, hydroxyfluorines, 3.9 up to 12-fold, and hydroxynaphthalenes, 2.9 up to 12-fold. We monitored, I can't even remember, I think it was 19 pH metabolites. Some don't go up because their primary excretion route is through the feces and not the urine, but the smaller molecular weight pHs, there's hydroxy metabolites that are easily found in the urine. So with respect to dermal contamination, we wanted to know if that's, in the literature, people have noted that dermal contact is a primary route of exposure to particle-bound toxicants like PAHs. So we were interested in looking at the relationship between what we see in the urine, so this is fold change urinary pH level, and what we find on the dermal surface. And you can see there's a significant empirical relationship, although there are some outliers, and it's interesting to note that these two outliers were individuals that spent an extended amount of time at a particular firefighting event. So essentially what we found here is that we know that the firefighters are wearing SCBAs because the workplace, the fire suppression scene is extremely hazardous. And so we expected that there would be a significant relationship between dermal contamination and internal dose. And moreover, we looked to see if there was any incidence or any evidence of lung injury. So we looked at Claricell protein 16 in the urine to see if there was any evidence of pulmonary injury, and there was none. So there was no change. And we found this relationship that I show here on the left. And this is consistent with numerous studies that have noted that dermal contact is definitely influential and can be more influential than inhalation. And in addition, we found excessive, we have a lot of sort of anecdotes, and we found an excessive level of urinary metabolites in an individual who forgot to wear a flash hood. So that's further evidence that dermal exposure can contribute to internal dose and urinary metabolite level. So what about this dermal exposure? What can we do about it? So, you know, we could say that firefighters should avoid initial exposure, but they're firefighters. So they're going to go to the scene of a fire, and they're going to be involved in fire suppression. So we can do things like improve occupational working conditions. So maybe there's a way to change the way firefighters, their behaviors at the firefighting scene. Or maybe we can improve PPE. So there are some efforts now to improve, for example, the flash hood. There's a new textile called Nomex that is supposed to be more protective. We can also do things like post-exposure dermal decontamination, and we've been interested in that a lot. And I'll show you, we've moved on from our first study to do a post-exposure dermal decon, as we call it, study to see if dermal decontamination actually works. So if you look online, or if you look in the literature, or you talk to people at firefighting conferences, etc., you'll hear a lot about wipes that are used for dermal decontamination after firefighting, and these are some of them. And the thing that really intrigued us is there's no scientific evidence that these actually work. There's claims from the manufacturer, for example here, uniquely formulated to remove the risk, but there is no scientific evidence that they actually remove any risk. What there is scientific evidence that you can wipe a PPE surface and remove something from the surface. Whether that contributes to reduction in internal dose, people don't know. So we moved on, and we started a study that we call dermal cleaning procedures to reduce firefighters' exposure to combustion-derived PAHs. Those are our primary substances of interest. And we worked in a training facility. So a live fire training facility. And this shows you here a firefighter. So I'll just back up a sec. This shows you a firefighter who's getting ready to go into this container where the training fire is lit. And you can see down here all the smoke that's emerging from this container. And this is a live fire training facility on Moody Drive here in Ottawa, where firefighter training is taking place. And this project is a project funded by a consortium of partners, including the Canadian Forces Fire Marshal. You can see different firefighter organizations in Ottawa and Montreal, INSPQ, Health Canada, and the IAFF, as well as the local union group. I think it's local, I want to say 162 in Ottawa, the local IAFF chapter. So essentially what we're interested in this project is we know there's dermal contamination because we measured it in our first study, and we know that there's internal dose. So we know that there's absorption, distribution, and metabolism, and then urinary excretion because we measured it in our first study. What we want to know is, can we engage in dermal surface decontamination and reduce the level of internal dose? That's really what we're interested in. Can we reduce the level of metabolites in the urine? So, and moreover, of course, we want to know if different strategies for dermal decontamination are more effective than other ones. So we want to do a comparison. So what we did in this particular study was we were looking at the efficacy of dermal decontamination to reduce exposure. We wanted to know how much is being removed from the surface. We wanted to know, does dermal decontamination reduce internal dose, the level of urinary metabolites? And I won't go into this. We wanted to know if dermal decon reduces DNA damage in urinary urothelial cells. That didn't really work. And then we wanted to know if some decontamination methods are better than others. So with respect to sample collection, here's the site again, the live fire training facility. And there it is again, a different shot of the trainees getting ready to go inside the containers. And so we did this in fall and spring, fall of 2018 and spring of 2019. And we looked at 3 dermal decon methods, commercial wipe A and B. To be honest, I don't know what they are. It's the only people who know are the person who did the actual decon and DND. DND knows. There was a soap and water dermal decon group, as well as the no intervention, no decon control. So with respect to sample collection, it's very similar to what we did before. We take a urine sample before, we take skin wipes before. At the firefighting event, so this is training now, we took personal air samples again. So same thing, we have the tube going into the pocket with the pump and the collection on the back of the neck. I didn't show it here again, but this time we actually used what's called, in addition, we used what's called a passive air sampler. So these are silicone wristbands, and they didn't necessarily wear it on their wrist. I'll show you, they wore it in different places. They wore it on their jacket and their wrist, and we used those, so that was on them when they went into the container to put out the fire, and we used that as a passive air sampling device. And that work was done exclusively by Health Canada, by Rocio Aranda Rodriguez. And then afterwards, we took the wipes again, much like we did in the first study. The difference here is that the individuals were then assigned to a decon group. So either no decon, soap and water, wipe A or wipe B. And then post decon, we took skin wipes again to see if the skin was clean. And then we took an integrated urine sample. And don't get confused by this. When we took the post-fire skin wipes, it was only from one side. And then those that went into the decon group, they used the decon method on the other side. And so we had wipe samples for post-fire and post-decon. Hopefully that makes sense. So I'm going to go through the results very quickly here. This is still a work in progress. The data analysis and interpretation is nearing completion. We hope everything to be published and wrapped up in the spring of 2022. With respect to air contamination, this is what a filter looks like from the personal air sampler that they wear on the back of their neck. So obviously there's very high levels of particulates. This is what a puff cartridge looks like. So here's these puff polyurethane foam cartridges in the air sampler before. These are unused. And this is what it looks like after a fire. And these capture, they're basically the lower molecular weight compounds that may not be absorbed to particulates and captured on the filter. So with respect to the level of contamination, our average total pH concentration in the air at the fire suppression scene in those containers was almost, well, above 350 microgram per cubic meter, up to almost 700. And as a point of comparison, the highest levels you'll see, for example, in urban Toronto would be in the 5 to 30 range. So of course, much, much, much higher, but not a surprise. I mean, this is a fire suppression scene. So what about skin surface wipes? What did we find on the skin surface? So here you can see this is the graduate student working on the project. She recruited some other students to help with the dermal wipes. We had a set template and a set method. So we did it for the firefighters. So it was always consistent. The methodology was always consistent. So with respect to what was on the dermal surface, if we ask the question, does the dermal surface become contaminated? Yes, it does. So we see an average of almost twofold increase in dermal contamination, total pH contamination at post-fire. So the maximum fold change we saw was over 25-fold. So there's a significant increase in dermal contamination after fire suppression. So no surprise there either. We had seen this before. But we wanted to ask the question, does decontamination effectively remove the pHs from the skin surface? And then secondarily, does the dermal decontamination reduce internal dose? So the level of urinary pH metabolites. So here is, I won't go through all this data. This shows you the post-decontamination changes in skin contamination level. So fold changes relative to post-fire. So the post-fire, after the fire, the skin is contaminated. And then we wanted to see, can these wipe methods remove what's on the skin? So what you see here, there's several compounds listed. What's important here is at the top, total PAHs, low molecular weight, and high molecular weight. And what you can see right away is for wipe A and wipe B, there's basically no change or no significant change. So they didn't work, except for this one instance here. They didn't really work. They didn't remove the PAHs that are on the surface. What worked was soap and water, and even then, it only removed about half. So it removed about half of the total PAHs, the low molecular weight PAHs, and the high molecular weight PAHs. And all these, where you see the asterisks, these are all significant changes. So the bottom line here is that we see, if we ask the question, does dermal decon remove PAHs from the dermal surface? Yes, it does, but only soap and water removed a significant amount of PAHs from the surface, and it removed about half of the dermally deposited PAHs. None of the other interventions significantly reduced dermal contamination, despite the manufacturer's claims. So we still have this remaining question about urinary pH metabolite levels and internal dose. So we wanted to look at whether the urinary pH metabolite levels are increased, so pre-fire relative to post-fire. And here you can see pH metabolite levels. This is the total, and this is what's called osmolality corrected. And you can see a significant 1.7-fold increase in urinary pH metabolite levels. So we found a significant increase in metabolites of pyrene, and you can see between 1.7 and 2.2-fold increase in these different metabolites in the urine: hydroxypyrene, phenanthrenes, fluorines, and naphthalenes. They were all statistically significant, and so there's significant evidence of internal dose. So none of this is a surprise, because we had seen this when we did our first study on emergency fire suppression. So to see it in a training facility is not a surprise, and people have published on this before. What we wanted to see is, does fire suppression increase the level of, well, so if we ask the question, does fire suppression increase the level of urinary pH metabolites? Yes, it does, as I just showed you, 1.8 to 2.2-fold increase in metabolite levels with a maximum fold change of 3.4. But then what we want to know is, did the dermal decon work? So even for soap and water, where we removed half of the surface, the dermally deposited PAHs, do we see a decline in the level of the increase in the level of pH metabolites in the urine? So what we found here is, no, it doesn't. So what we see here is, this is the fold change in urinary pH metabolites for the different intervention groups. And this is showing the 1.7-fold post versus pre increase in pH metabolite levels. So what I showed you on the previous slide. So there was that 1.7-fold increase post versus pre, and what we found here is that there's a 1.7-fold increase right across the board, so that the wipes didn't change that. So essentially, if we ask the question, does dermal decontamination reduce internal dose? What we found is none of the interventions significantly reduced the observed fold change increase in urinary pH metabolite levels. So it was never significantly below this observed 1.7-fold. So we saw an increase. None of the interventions changed that increase, not even soap and water. So, I mean, the question here, oh, well, I'll move on to what we think is going on here in a moment. But first, I wanted to talk to you briefly about the silicone wristband results. So the results available to date. So what we've been doing here, this is a bit of a side project. We've been trying to see if silicone wristbands are useful as a passive air sampling device in occupational settings. People have looked at this a bit, and there's a literature on this, but we wanted to see at firefighter training, for firefighter training activities, and now, as I'll show you for our current work, we wanted to see about emergency fire suppression air sampling. So what we looked at is the relationship between what we see on the wristbands and what we see in the active air samplers. So we saw a significant relationship between silicone wristband jacket values and active air values. And jacket values, when I say jacket values, it's the results of the silicone wristbands hung on the jacket and versus the silicone wristband that we put on the wrists. So we see a significant relationship between their jacket values and the active air values, and the jacket values and some urinary metabolites. This was all work done by Health Canada, as I mentioned, work by Rocio Aranda Rodriguez. And we didn't see a relationship between the wrist values and the urinary metabolite values. And what we think there, what's going on there is that the silicone wristband on the wrist was covered by a nitrile glove or their turnout gear glove, so it wasn't significantly exposed. And this plot here, I'm just going to show you the relationship that we see between what we observe on the concentration in the silicone wristbands in nanogram per gram of wristband against what we see in the active air sampler results. And one of the reasons why this is important is because collection of active air samplers with a tube behind the neck and a pump in the pocket is not easy. It's not easy on anyone. It's difficult for us to organize, and it's difficult for the firefighters because they have things they need to do when they're involved in emergency fire suppression. So we see here, if you look for these different pH metabolites, I'll just click through it very quickly. We see a significant relationship between what we see on the wristbands and what we see in the results from the active air sampler. So this is very encouraging, and we're moving forward with a project now that I'll talk about in a second, using the silicone wristbands. And this is a significant relationship, statistically significant. So if I summarize the dermal decontamination study findings, what we found is that the training environment is heavily contaminated with airborne PAHs. There's a significant dermal deposition of PAHs. Aerial dermal PAH level is significantly reduced by soap and water. So it works, the soap and water works, but what doesn't work is the decrease in internal dose. We found a significant post-fire increase in urinary pH metabolite levels, but none of the dermal decontamination strategies reduce the post-fire increases in urinary pH metabolite levels. So it doesn't reduce internal dose. We also saw, as I showed you on the previous slide, that silicone wristbands are useful for passive sampling. And what's important here is that we're currently doing some modeling to figure out what might be going on. So what we think is that the penetration and distribution of the dermally deposited PAHs is happening too quickly. So the penetration into the skin and into the bloodstream for distribution and metabolism, we think, based on our modeling up until now, is happening in less than 10 minutes. So if they're in the container for 90 minutes, and then we wipe them down and they deliver a urine sample 18 hours after, it's much too late. So one thing I wanted to say just before I move on is none of this could have happened without Jennifer Keir. She's the PhD student working on this project, and essentially she's been excellent, amazing. She's organized all of this. She's done all the work on the first study, all the work on the dermal decon study. You can see her here helping one of the subjects put with the pump, make sure the pump is working properly. So I'm just going to move on to talk about the other work that we're involved in. So there are other collaborative studies. This slide is telling you about a collaborative study led by McGill University. And this is Keratone. Keratone is working with Jonathan Chevrier, and they're working on a project that involves McGill, INSPQ, Health Canada, and the Montreal Fire Service. And it's a pilot study investigating Montreal firefighters' exposure to selected chemicals. And the chemicals that we're looking at, or that Keratone is looking at, is levels of Pfas, which are per and polyfluorinated alkyl substances in serum, blood serum, as well as PCBs and brominated flame retardants, dioxins, furans, and heavy metals. And then in addition, at Health Canada, so that's being done in INSPQ in Quebec City, and in Health Canada, Keratone is looking at using urinary biomonitoring to look at organophosphate ester flame retardants or plasticizers, and polycyclic aromatic hydrocarbons and bisphenols. So a wide range of substances that they're looking at, but we don't have the results yet. The results will be published at the end of, or the results will be interpreted and published at the end of 2022, following completion of the data acquisition, data analysis, interpretation, and importantly, communication to the study subjects. So with respect to, so what I showed you before, the two studies that I showed you at the beginning, the initial one funded by the Ontario Ministry of Labour and the later dermal decon study, primarily funded by the Canadian Forces, those preceded what's called the Government of Canada's Firefighter Action Plan. So now the Government of Canada has been concerned with the exposure of firefighters to harmful chemicals and has been seriously engaged in promoting research in this area. So the government formally announced a firefighter action plan. Here you can see the news release was announced by Environment and Climate Change Canada and Health Canada. Moreover, in the Liberal platform statement, Forward for Everyone, there's actually a statement, a formal statement about a comprehensive action plan to protect Canadians, including firefighters. So in that firefighter action plan, going very quickly, there's some short-term and long-term actions that we're involved in. And when I say we, for example, Rocio and I, and one of, as well as people in the Safe Environment Directorate of Health Canada, we're involved in collection and analysis, curation and analysis of published human biomonitoring and environmental contamination data. And these analyses are currently underway. And this is thousands of observations from the literature. We basically wanted to see what's the state of the knowledge on firefighter exposures to toxicants of concerns. There's also activities in research, monitoring, and surveillance, both short-term and long-term. Short-term actions is to finish what we're doing now. So to finish this McGill Montreal Fire Service collaborative work that I just talked about, as well as this University of Ottawa, Ottawa Fire Services, Canadian Forces project, the Dermal Decon project. The long-term action, and I'll talk to you about this in a tiny bit, in a few slides, is to undertake new collaborative biomonitoring initiatives. And for example, there's an HC-led study, Health Canada-led study, to assess firefighters' exposure to brominated flame retardants and Pfas during emergency on-shift suppression of municipal structural fires. So very similar to our first study, but the first study was only on PAHs. Now we're moving on to Pfas and flame retardants.
Well,
just very quickly to show you a few snapshots of what we've been doing, what's coming out of our literature review. So these are some figures that were prepared by Rocio, so showing you.
Paul, I just want to make sure I know you're almost done. I want to make sure there's a little bit of room for questions. So just
yeah, I'm almost done. So I'll go through this very quickly. What we've been doing is looking at comparing the level of contaminants, in this case PBDE flame retardants, in fire hall dust. So those are total PBDEs in fire hall dust, and you can see compared to residential dust, this is a waste electronic and electrical recycling facility. So very heavy contamination at fire stations, fire hall dust, and a waste recycling facility compared to residential dust. There's for only 5 PBDEs compared to total PBDEs. And I'm actually going to skip this slide. because I'm running out of time. It's just another plot showing some data looking at fire hall dust relative to dust collected in residential homes. And the last thing I wanted to tell you about, I think this is my second to last slide, is that we're currently engaging in a formal study to look at firefighter exposures to flame retardants and Pfas during emergency on-shift fire suppression. So we're using these silicone wristband passive samplers to monitor at the fire suppression scene, in the fire hall, and in the truck cabs. And we're also collecting additional information from looking at fire hall dust, and we're analyzing the dust and silicone wristband for an extensive array of contaminants, toxicants of concern. And so those results will form the basis of a follow-up biomonitoring study to assess internal dose of those toxicants of concern. And this is all very consistent with the Government of Canada's Firefighter Action Plan and concerns of the IAFF, and we'll communicate the results to a wide range of stakeholders. And that's it. I'm just going to wrap up with some very brief concluding remarks. Firefighters involved in emergency suppression of municipal structural fires are significantly exposed to combustion-derived carcinogens. The results confirm that dermal contact is an important route of exposure. Dermal decontamination study shows that we can remove half of the dermally deposited PAHs, but we can't, dermal decon doesn't affect the internal dose. The urinary metabolite level did not change. The silicone wristbands were useful for passive exposure monitoring, and we're going to continue to use those in our current study that's going on now. The action plan has numerous activities, including extensive literature review to assess the state of the knowledge about firefighter exposures to toxicants of concern. Our current CMP-funded work, as I mentioned, is looking at those toxicants at the emergency on-shift fire suppression scene. And our ongoing work, just to wrap up, our ongoing work basically provides foundational information that we can use to design and implement remedial measures. I mean, that's what this is all about. We're hoping that we'll be able to provide advice that can be used to minimize exposure and health risk. For example, improved PPE, new SOPs, etc. And I'll wrap up there.
Well, thanks. And just to tie this together, for if people are looking at videos from our previous October sessions, and I know you have some colleagues on with us as well, Paul, so thank you. This relates, this subject matter, obviously, to occupational, and we just had this technical discussion at our team meeting this week around looking at the wipes and whether they actually decontaminate. We were talking about heavy metals. You were talking about PAHs, and there may be discrepancy between those, so it's super interesting. But to tie together, for previous October sessions, we had the Health Canada folks come and talk about the chemical management plan, and this is a project related, like, that spawns from that. So we can see the progression of that and how important it is for us in Ontario to be constantly interrelated with as many partners as we can. Paul, thank you. And I know Sarah's, I saw Sarah on the line, and we've been doing a project with Health Canada looking at the environmental risk assessments compared to the occupational control banding risk assessments, and us working together and collaborating is all what this whole thing is about. So I really appreciate. Do you have occupational hygienists on your team, Paul?
Yeah, we absolutely have to have one. The Research Ethics Board required that, and it's Tracy Kirkham from the School of Public Health at the University of Toronto.
Great, wonderful. Yeah, because I started into this thought process in my whole background world, taking environmental toxicology courses in my undergrad, and then didn't even hear about what an occupational hygienist was until I finished all that. So it's just amazing work. I do have a couple of questions in the chat, and it's okay, Nancy and I can shorten our little blurb because this is also fascinating. But Dave is asking, what about inhalation exposure when respiratory protection is removed? I know you.
Yeah, I mean, that's a good question. I mean, obviously you wouldn't remove respiratory protection during attack and knockdown, but you might remove it if you're at the truck site or if you're involved in overhaul. So we didn't look at that specifically, but some people have, and they found, for example, at overhaul, if you compare the people wearing SCBA with those who do not, you see significant increase in exposure, presumably by inhalation. So it's a complex issue, the route of exposure and the degree to which the PPE offers sufficient protection, but there is evidence that the SCBA works during overhaul.
And yeah, so thank you. And what we recognized during COVID, again, with this collaboration, is that we need lots of disciplines working together and sharing language and crossing over boundaries. And I see, it's important here with the chemical management plan. Yes, you're looking at administrative controls for firefighters, but you're also looking at controlling what we put into furniture.
Yeah.
So that we, right cradle to grave. That's why it's
Yeah.
so important to have environmental folks. And, we get very narrow in our, insular in our study and in our, what we're considering. And it's so really great to involve Health Canada, who are also looking, cradle to grave, why are we making furniture that has that foam in it if that's, causing, just as an example.
Yeah, and I didn't even have a chance to mention that. I wasn't going to mention it because it's not my area of expertise.
Right.
But the firefighter action plan includes those actions related to those concerns. So product safety, for example.
Exactly. Thank you. And Emily has a question, and then I think we'll have to move on if that's okay. But rest assured that the video is going to be there for people in slides. Paula does have to check with Health Canada about a PDF of the slides being shared. We do that if we can, but we'll wait respectfully for the answer. Have you considered also doing serum sampling for PFAS, or will you be focusing on urine sampling?
So in our current study that's going on for PFAS and flame retardants, we're only doing the passive sampling using the silicone wristbands. And the reason is we want to see if we detect those agents at the scene of emergency fire suppression and what the levels are. So we're doing it in a staged fashion. Our first stage is determine if they're there and what the levels are, and then determine what would be an effective follow-up study, whether we look at serum or urine or whatever. We'll probably do both if we have sufficient funding. The funding is always an issue, that's for sure. But good question.
Thank you so much. And Paul, that's where we're going to have to switch gears today. So again, Thank you so much for coming, and stay tuned. We'll, we're definitely going to reach from the OC disease action plan or OC illness prevention. That's what we're about to talk next. We don't have a cool term yet, but we really want to continue involving Health Canada and collaborating together. And thanks for your attendance and your sharing of your expertise today. So thank you so much.